So I am going to be talking to you tonight about leafy prairie clover, Dahlia foliosa. Many of you might be familiar with this plant. It's a federally endangered species known from Illinois, but much of my work is focused on Dahlia in other parts of its range. And so I'm going to talk a little bit about limestone glades, which is the habitat that I know it from. And then I'm going to talk about Dahlia more specifically. So first, give you some ideas about major research themes in my lab. As Kurt said, I've moved around a lot, but my interests haven't really changed that much over the years. So I've always been interested in conservation, trying to focus my research on a project that's going to have meaning for some sort of on-the-ground effort. I've also been interested in reproductive ecology. Specifically, I'm interested in clonal plants, so plants that reproduce asexually by root sprouting, for example. Chris mentioned he's a forester. I'm a big time tree hugger by trade, so I'll talk a little bit about that in just a minute. I'm also interested in biogeography. As I said, I'm interested in movement of plants over time in space. And so all of those things, those themes sort of show up over and over again in the projects that I select with my students. So my work often has a field component. This photo that I'm showing here is actually from a cedar glade in Middle Tennessee. The woman leaning over the plot is one of my undergraduate students from Middle Tennessee. And the man just outside the plot is Dr. Matthew Albrecht from the Missouri Botanical Garden. This was an effort when we were doing demographic monitoring of a federally endangered plant called Pine's ground plum that's only known from Middle Tennessee. And then this image shows you the other aspect of my work, which is lab genetic work. And so unfortunately these days, I feel like there's more, there should be another picture here of me sitting in front of a computer in an office because that's where most of my work takes place. But I do get to work with students who are able to, gain experience in the field and in the lab. Okay, so what species do we work on? As Kurt mentioned, we've worked on a lot of different things over the years. So I'm just going to tell you about the ones that we're going to be working on this summer. I've only been at Furman. I'm finishing up my second year at Furman and moving to a new university right before COVID is a really fascinating way to start an academic program. So my lab is really just getting going here. But this summer, I've got students who are going to be working on a number of different projects, one of which is Dahlia foliosa, which I'm going to be talking about tonight. But with Dahlia, we have continuing work through U.S. Fish and Wildlife funding to the Tennessee Department of Environment and Conservation, who I'm collaborating with. We'll be working, I'll talk more about that in a bit. American beech is, if I had to pick a favorite plant, it would be American beech, Chris, because this was my first love. And I'm interested in clonal population genetics in the Smokies in particular. And I have a student who will be working on that this summer. I'm starting some new work in the federally endangered bunched arrowhead, which is only known from Greenville County, South Carolina, where I am, and one or two adjacent counties in North Carolina. This is a semi-aquatic plant that also reproduces clonally. So you can see, again, some consistency and themes here. And then finally, I'll have a student working on Oconee bells, and there's no flower here because I just took this picture a few weeks ago, and they are apparently in flower right now. So I've got to get home from Alabama where I'm visiting my mom so I can see Oconee bells in flower. This is another rare species. It's found in South Carolina and North Carolina, and I have a student who's again going to be looking at clonal structure in this species. So lots of interesting things going on for us this year. But what I'm going to talk about tonight has to do with work I've been doing for a number of years, and this work has largely taken place in Middle Tennessee. So the photo that you see here is just an absolutely gorgeous barren, is what we would call it. It probably resembles the prairies that you guys have in Illinois, but this is a grassland habitat that we see associated with limestone glades in the Middle Tennessee area. So this is a photo that I took at Flat Rock Cedar Glades and Barrens State Natural Area in Murfreesboro, Tennessee, just a few miles from the Middle Tennessee State University campus. And the plants you can see there are, I believe that's Echinacea simulata in the front, and then in the yellow that you see in the back, that's Ratibida pinnata. And that, I'll say that image was taken within, I think that was a year after a burn. And so a really spectacular year for the coneflowers. So what exactly is a limestone glade? The map that you see here is taken from Baskin and Baskin, 1986, and the shapes that you see on the map are indicating localities where they had identified glade habitats. So the way they defined glades at that time were natural treeless areas characterized by limestone or dolomite bedrock. So there's a geology component that's particularly important here. They're dominated by herbaceous species, but there are often some trees, particularly in this case, we often see cedar trees, which is why some people have referred to these as cedar glades. The greatest concentration of these habitats is found in the central basin of Tennessee, which is in that Middle Tennessee region where you see most of the dots there. So there have been more than 400 species of vascular plants documented in these glade habitats, and at least 29 are thought to be endemic to those sites, meaning that they're not found outside of those types of habitats. So pretty spectacular floristic diversity, some really interesting things that we see there. The other thing about glades is they're weird. If you've never visited a glade, most people, the average person might look at a glade and not think much about it because they're rocky in the summer. They're extremely dry and extremely hot. I'm talking really hot. In the spring, you'll often see ephemeral streams in places that weren't wet before. And so a lot of people in Middle Tennessee have used these as dumping grounds. You'll find lots of areas with huge trash piles because they weren't really deemed as good for anything else. They also weren't really thought to be very good for agriculture or for other kinds of development, which in some ways has been a good thing for the preservation of the plants present in these sites, because they're in a region where there's been a lot of development. So Middle Tennessee is apparently one of the fastest growing regions in the country. Just the Nashville metro area and just outside of Nashville metro has grew dramatically just in the time that I was there. And as a result, you tend to lose a lot of habitat that way. So to show lots of pretty flower pictures, here's some examples of, I took all of these at the Flat Rock Cedar Glades and Barrens site that I mentioned before. You can see a lot of cool things that we get to see at these sites. So here's that Echinacea simulata again and Ratibida pinnata. Those were the two in the coneflower photo that I showed. Another species that I've studied in the past, Astragalus bibulatus, only known from a few counties in Middle Tennessee. We just published a paper on that that came out just this past weekend, so pretty excited about that. Relative Astragalus tennesseeensis is also found, and interestingly, if you're not familiar with this species, it has a similar distribution to Dahlia foliosa that I'm going to talk about a little bit later. A few others, folks are seeing Leavenworthia stylosa right now. It's one of the first things to flower on the glades, and so you'll see loads of photos of Leavenworthia at this time of year when there's not much else out there. One of my personal favorites, Matelia carolinensis. I think this is just such a cool viney thing. At any rate, so many beautiful plants that you see in the glades. So something that I've been interested in for a long time is the evolutionary origin of these glades, meaning why are they here? Why do we have this patchwork of small, isolated, and fragmented habitats with these unusual floristic communities? And so this map is just indicating sort of a cartoonish way of looking at the distribution of glades and glade-like areas. And so you see you have these Middle Tennessee glades and surrounding areas, but then you've also got some in Arkansas and in Texas that some of you may have heard about. So we're really interested, I say we because I'm referring to my colleagues and myself, we're really interested in understanding how these habitats came about, at what time in evolutionary history they may have come about, where did they come from, what drove these isolated species into the places that they're in now. So that's going back to that idea that I'm interested in biogeography in addition to some of those other things that we talked about at the beginning. So I'm going to say this is like assembling the Avengers. I've put together a team of what I think are pretty spectacular folks, and we've dubbed ourselves the Glades Gang. This is our research team that's trying to look at some of the big picture questions about these evolutionary origin stories, if you will, of the cedar glades. So you may or may not know these folks, but they're from all over, as you can see here, and I'm excited to be working with them on some things. Clayton, you'll see his name come up again later in the talk because he and I are working together right now on some Dahlia foliosa work. The rest of this team, we're in the early stages of developing a proposal for a big picture story about cedar glade origins or limestone glade origins. So our idea is that clades may hold the answer. Well, what does that mean? So let's take a look. We're interested in species that are endemic or nearly endemic to glades in the Middle Tennessee area that have their closest relatives in as widespread species in the Midwest. And so our hope is that by looking at a number of different species that meet this pattern, we'll use a bunch of different genetic tools to try to identify what drove the floristic communities that we see today in these glades. So examples of species that we're targeting for this work, of course, Dahlia foliosa, also Pine's ground plum that I mentioned a few moments ago. Nashville breadroot, Pediumillum subacali. All three of these are actually legumes. They're all in the family Fabaceae. And then we're also going to be looking at echinacea. This is Tennessee coneflower, Echinacea tennesseensis. So all of these taxa are somewhat restricted in their species distributions, but are thought to have their closest relatives in the Midwest. So some interesting things to look forward to in the future of this work. Tonight, though, I'm going to focus specifically on the population genetic work that my students and I and some collaborators have been doing in Dahlia foliosa. So if you're not familiar with Dahlia foliosa, I'll just point out that it has a really odd geographic disjunction. And so the three circles here represent the three disjunct regions of the species range. So let's see here. You have the Tennessee limestone glades in Middle Tennessee, and that's where the bulk of the known populations are. The Alabama limestone glades in northern Alabama, there are only a handful of populations there. In fact, depending on who you ask, there may be as few as 4 populations in that area. And then the one you might be familiar with is the Illinois Dolomite prairies. So there are there are more populations in that area than in the Alabama region, but still far fewer than what we see in Tennessee. I will point out that Illinois has had a really phenomenal history of managing these populations with reintroduction efforts and augmentation efforts. And some of our work has actually focused on population genetic structure of those reintroduced populations relative to the natural ones. I'm not going to have much time to talk about that tonight, and that's still developing in terms of analyses and those kinds of things, but I can tell you a little bit about what we found. So what are the questions that my collaborative team has been asking over the last several years? We're interested in how genetic variation is structured within and among disjunct regions, meaning how similar, how different are Illinois, Tennessee, and Alabama populations. We're interested in reintroduction. This is something we looked at with Astragalus bibulatus. It's something we're looking at now with Dahlia. But we want to be able to use genetic information to say something about reintroduction efforts and future reintroduction potential. And then finally, what can genetic data tell us about the implications for conservation management? So I'm going to show you a series of figures from genetic analyses, but I'm going to give you sort of the take-home points of each figure. So don't worry if you feel like there's too much going on there, but hopefully I'll be able to explain things in a way that makes sense. The key finding for us is that the genetic clusters that we have identified are not consistent with the geographic clusters. So Tennessee, Alabama, and Illinois do not equate to independent genetic groupings. Okay, so the image that you see here is something we call a structure plot. And the main thing to focus on here is the state designations. So the Illinois populations, there are 4 here in this analysis. The Tennessee populations are here in the center. And then in Alabama, we have 4 populations as well. So then the other thing you want to look at is color coding. So color indicates genetic similarity. We'll just leave it at that for our purposes. This particular analysis identified 3 genetic clusters based on all of the populations that we sampled across these 3 regions. And so what you can see is Illinois is in this blue cluster, but you also see that light blue cluster showing up in Tennessee populations. Alabama is largely in this green cluster. You can see some of that showing up in Tennessee as well. And then Tennessee has this dark blue cluster that pops up in Alabama. So the key here is that we have 3 genetic clusters that do not correspond cleanly to the 3 geographic regions, which is kind of interesting when you think about it. There's a pretty significant geographic break between the Tennessee and Illinois populations, and you would really expect those to be more different from one another than they are, based on the assumption that there's not been gene flow, not been pollen exchange, and things like that between those regions. But that's not what we found. So interestingly, what we do see is that the Illinois populations are actually quite similar to the Tennessee populations based on the genetic data that we collected. And you can, again, see that through this light blue cluster assignment that shows up again in several Tennessee populations. This principal components analysis is just another way to visualize this. The main thing that I want you to take from this is that you can see a great deal of overlap in the genetic profile of Illinois populations and Tennessee populations. Notice that Alabama populations are a little bit more distinct, but that could be a consequence of how few individuals we were able to sample at those sites. The key here, though, is this overlap in genetic patterning between Illinois and Tennessee. That's going to be important for a point that I make in just a few minutes. So my colleague, Clayton Visker, has done some really cool environmental analyses. We had planned to do some environmental niche modeling, which is an approach where you take occurrence data and you take environmental data layers and you model what the supposed niche or place of inhabitation of the species is. With rare plants, it's kind of difficult to do that because you don't have sufficient sample size from a statistical perspective to get a strong model. So Clayton took a different approach. And what he found is that Illinois populations occupy a unique environmental niche relative to Tennessee and Alabama. Well, what does that mean? It kind of makes sense when you think about the fact that I mentioned in the beginning that the Illinois sites are these dolomite prairies, and the Alabama and Tennessee sites are limestone glades and barrens. The figure that I'm showing you here is a principal component analysis of environmental data. So this is not genetic at all, okay? And the way that he's analyzed these data, the solid ellipses represent the current environmental space that a given group of populations occupies. So in this figure, Tennessee is in gray here, Alabama is in red here, and Illinois is way over here in yellow. So what that tells you is the environmental space that Illinois populations occupy is very different from that of Tennessee or Alabama, and that Tennessee and Alabama share a lot of the same environmental space. The other ellipses that you see here, the ones with the hash marks around the dotted edge, represent future predicted environmental space based on climate change models. And so what we can see is that you do see a shift in predicted environmental space for all the populations. But again, Illinois is over here, separate from Tennessee and Alabama. So again, Alabama and Tennessee share space. Illinois occupies a completely unique environmental space. So what implications does all of this have for, I was checking the chat there. I see a question. I'll follow up with that in just a second. Implications for conservation and recovery. First of all, we did observe that Dahlia foliosa has lower than expected genetic diversity based on its range and based on its life history strategy. And this has been shown before in a previous study. Our data suggests a really complex pattern of genetic structure that does not reflect the geographic pattern that you have observed, which really complicates the way that we think about management units from the perspective of conservation and recovery. So one might be tempted to use genetic clusters as a thought process behind management. However, as I showed you, our environmental analyses indicate a very different pattern than the genetic analyses. So while genetics indicate Alabama to be somewhat distinct and Tennessee and Illinois to be more similar to one another, environmental analyses show significant niche differentiation across the range, with Illinois being different from the other two. And so when you take all of that into consideration, you kind of have to consider a multifactorial approach to conservation strategy. So some things that Clayton and I have been talking about. First of all, we definitely think there needs to be some formal demographic monitoring in Alabama. Right now, Alabama is the only area that is not doing annual monitoring on Dahlia, at least not to the same extent that Tennessee and Illinois have done. But the other big thing that we think could be really interesting and informative for the species is to think about common garden experiments where you take plants from different parts of the range and plant them in similar environments, evaluate how they respond to that environment, and that helps you to better understand whether or not there's been some sort of physiological adaptation to different regions for the same species. And why would we do that? That's important to think about future reintroduction efforts. Maybe we have genetic similarity between Tennessee and Illinois, but maybe there are adaptive conditions there that we can't visualize in the data that we have. And if we were to start moving plants between the two regions, it could actually be detrimental to the species rather than helping the species. So there's a lot to consider there. And we're in the early stages of thinking about what this might look like. So we're doing a number of other things in this system. As I mentioned, I'm working with folks in Tennessee this summer, and we're actually going to, my small part of the larger project is to use genetic data to try to help define element occurrences for the purposes of management. I have a student that's done some work looking at variation in the number of seeds produced per seed head at different sites in Tennessee. And the other aspect of this work that I mentioned earlier is we do have data on genetic variation within and among sites in Illinois, both in natural populations and in reintroduction sites. And one of the really cool things that we're seeing there is that for reintroduced sites, which are typically using multiple source populations for seed source, you're actually seeing more genetic diversity in those sites than in individual natural sites. So some exciting potential there for the future of the species. With that, I just want to acknowledge that this is a team effort. I did not do all this by myself. A number of folks were involved in project development and discussion. Jeff Call, my contact at U.S. Fish and Wildlife in Tennessee, initiated this work with me and was integral in discussing strategies for sampling. Kathy Pollock in Illinois was a huge help in terms of acquiring material from Illinois, providing information about management in the region, and providing some feedback on an initial draft of a manuscript. And then, of course, Clayton has joined me on the environmental work that I mentioned just a bit ago. A number of undergraduate students worked on this project, and a lot of people at the state and federal levels worked on material collection in the field. So a lot of people contributed to this effort.